All right, I hope you can hear me okay. So thank you very much for the invitation and the opportunity to present some of our data today. I come from Cytiva, and we provide end-to-end solutions for manufacturing of a lot of different biomolecules. And this involves hardware, software, and consumables. What I show you here is the complete solution for mAbs. So we have everything from the upstream to the capture, polishing, during the downstream, all the way to fill and finish for the final drug substance. This, of course, involves a lot of work, and it involves a lot of analytics. So I will present a few cases throughout my talk, and we will jump right into the first case. This is characterization of a mAb culture over 14 days. I know that this is a very busy slide, so I will walk you through it. What I show here to the left is that we cultured for 14 days and sampled every day. Then on an ECTA, we did capture with protein A, and then a desalting step. So we have, now it's not really visible, sorry, but we have a elution peak and a desalting peak. Based on the UV, we can get the concentration and the yield. So we see that throughout the cultivation, we have increased concentration of our antibody. Then I'll show you two standard chromatography methods to assess maybe the most fundamental critical quality attributes. In the middle is the SEC. So here we have high molecular weight or aggregates, eluting before the monomer, and then we have later eluting low molecular weight or fragments. In the middle at the bottom here, I show you then the purity profile throughout these 14 days. And this is a very stable process because we have less than 1% aggregate every day. So it's very pure. At the right, I show catalytic or analytical cationic exchange chromatography to assess charge variants. Here we have a main peak. Everything eluting before is acidic variants. Everything eluting later are basic variants. And what I show you here at the bottom, the green are the main peaks. So we can see that the main peak is dropping down after roughly 9 days. And at the same time, the acidic peak is increasing, while the basic are continuously going down throughout the process. So we have a shift here in variance after roughly day 9. So these are what I like to call standard chromatography methods, or basic, but we need advanced analytics are required for deeper characterization of our antibody. And this is where we turn to mass spec. So we have a few BioAccord systems at Cytiva, and they are multi-purpose, so we run a lot of different biomolecules on the same systems. What I show on this slide is that I've grouped them into proteins, nucleic acid, and then other. And I hope you can appreciate that we have a lot of different separation techniques. And then I've also listed here the different demands that we have on site. So we cover a lot of different biomolecules, a lot of different characterization, but most of the time we're running intact mass analysis for proteins and nucleic acid, but we can also do like peptide MAM or peptide mapping. So if we go into some example of analysis, this is intact mass analysis on the BioAccord. So here we have the UV, we have the tick, and then I've just extracted the tick at 3 time points. You can see the spectra in the middle, and then the deconvoluted spectra to neutral masses at the right. So this is just a reference mAb. We can see that we have light chain, half antibody, and then intact antibody. And it looks very pure. If we go back to our case 1, where we monitored a culture for 14 days, I had selected a few days here from early to late just to see what is happening with our mAb with extended cultivation time. So we can see to the left is the UV, and to the right is the tick. We can see from the start that we have very little light chain that seems to increase with abundance. And then we can also see that we have half antibody and full antibody. The tick will be more sensitive, so it looks more complex than the UV. And what we can say from this, I'm not going to go through all the molecular weights, but I hope you can see that the complexity of the culture increases with cultivation time. So we have more species with time. This is now the same samples, but here we have digested them. So we do subunit analysis. We do it with IDS, so we chop off, and we have the Fab prime and FC part, and then we also reduce it with DTT. So we will have 3 peaks here. It's the FC half, it's the light chain, and then the FD prime. Again, UV and tick. And I hope you can see that going from day 3 to day 14, we're going to get more peaks. It's going to be a forest of peaks, so we're going to have more species, more modified variants with extended culture time. So by doing subunit analysis compared to intact mass, we can start pinpointing where are the modifications occurring on our antibody. This is another type of analysis on the same samples. So gaining more information about the glycans, we chop them off, label them, and then we do mass spec on them. So what I show you here is just the glycan performance test standard. To the left, it was injected in the start, in the middle, and at the end of the sample list, and I hope you can see that it's identical throughout. To the right are the MAB samples that we followed for 14 days. And the glycan profile is slightly shifting throughout the cultivation, but it's kind of stable, so it looks good. So these were some examples of more traditional or easy antibodies. Now I'll turn to more challenging or newer antibodies, which are then bispecific antibodies. And why are they more complicated? That's because if we look at the IgG-based bispecific antibodies, they will be composed of 2 different heavy chains and 2 different light chains. The desired species should contain then blue and green light chain, blue and orange heavy chains. But since these are co-expressed and then formed, we will have a lot of different impurities or potential impurities. We can have the heterodimeric Fc species. So these have the correct blue and orange heavy chains, but the light chains are swapped. We can also have homodimeric species. So these have the same heavy chain. So it's blue-blue, orange-orange, and then different light chains. And then we can also have half antibodies. And of course, we will have aggregates, and we will have smaller fragments. So you can envision that we will have a lot of different impurities that we normally don't see with a traditional MAB. This places a high demand on both manufacturing, but also analytics. So to purify them, we need selective resins, and to analyze them, we need accurate analytics. So now we'll move over to case 2. That is a bispecific antibody that we call BSAB1. I would say this is a pretty challenging antibody because it has the structure of a traditional IgG, and it's very similar masses. The heavy chains are a few hundred Dalton differences. The light chains are 31 Dalton differences. So it's not much to play with the sizes here. And as I showed you on the previous slide, we have many different impurities, at least in theory. So if we look at the structure, this one will bind 2 different antigens, black, green. It will have 2 light chains that are of different isotypes, lambda, kappa, and then we have the Fc part. And what I've highlighted here are 2 of our capturing resins. It's a MabSelect Prisma. This is a protein A-based resin. And I have MabSelect VL, which is a protein L-based resin. Prisma will bind to the Fc part, so down here on the antibody. MabSelect VL will bind specifically to the kappa light chain, not to the lambda light chain. If we then take our start material, we know or we think we're going to have a lot of different impurities, and then we do 2 straightforward purifications. We use our protein A-based and our protein L-based. We just load our sample, lower the pH, and then we will elute everything in one step peak here, highlighted in blue. We collect that peak, and then we run a lot of different analytics. I'm not going to go through everything, but if we look on the SEC, the purity, we can see that we have pretty okay purity for just the capturing step. But we can see that we have more low molecular weights with the protein L-based resin. Not surprising, because this will capture the light chain. If we look on the mass spec, we can really detail what are these different species. We can see that we have presence of half antibody and full antibody. In the VL Elouette, we can also see that we have LC dimer and 2 LC alone. So here we characterize the start material, and then we aimed for a second polishing step to see if we can purify our desired product out of this more or less soup. So we know that we are going to have our correct antibody, but we could also see in the islets on the previous slide that we have a mismatch of the light chains. So the one we have is going to be the one in the middle, black and green light chain, black and green heavy chain. And here we separated on an ion exchanger, so cat ion exchanger, CAPTO-SP Impress. We loaded the Prisma eluate from the previous slide, and then we increase the salt concentration. So we have a salt gradient, and then we will have 3 distinct peaks and 1 shoulder. We collected these, so we fractionated the peaks, and then we did analysis on them. And then we could see that we can separate out these different 2 main impurities from the correct desired full antibody. So peak 2 is the one that we want. Peak 1 and peak 3 we don't want. So if we look at the mass spec data for this, to the left is the MabSelect Prisma eluate. So this is just where we bind, elute, and then we will have these 3 species within the same spectra. Remember, it's only 31 Dalton difference, so this is pretty challenging to pick out because we will have the different glycoforms as well. If we take the peaks from the CAPTO-SP Impress, so the cation exchanger, we can see that we have wrong, we have correct in peak 2, and then we have wrong based on our mass spec data. So this really helped us to see where is our correct antibody in the process. For anyone paying close attention, you can see that, okay, we now have black and green heavy chain, black and green light chain, but the light chain can just be swapped and we will have the exact same molecular weight. And that we can't see on the mass spec. Luckily, we can do that with Biacore. So here we checked what we call bispecificity using Biacore SPR system. We have on the surface, we have immobilized our first antigen, A1 in black. We take our sample and flow it over. If it has the black-black composition, it will bind to the antigen. We then take our next antigen, flow that over the surface. If the one antibody on the surface has then green-green on the other side, it's going to bind the second antigen. By doing this, we get 2 different signals. The first one we call a capture signal that we can quantify. Second signal, we get an enhancement signal that we can also quantify. Taking the ratio, enhancement with capture, we get a bispecificity value. This should be 100%. Then we have everything that binds the first antigen will also bind the second antigen. Starting material was 70%. Our peak 2 from the CAPTO-SP Impress was above 95% in bispecificity. So not only is it correct mass, it's also binding correct antigens. Now we'll move over to case 3, which is the second bispecific antibody. This one is a little bit easier, at least in terms of structure. The left part in black is similar to half an IgG, so it has a kappa light chain and then a heavy chain. The right part in orange is a heavy chain, single chain FV. This is then one chain that has element of an FC region from an IgG. It then has a heavy chain that is VH3 isotype, and then it loops back and has a part that is similar to a lambda light chain. But it's all expressed as a single chain. And here we have a lot of possibilities to use our resins to capture. So I've selected MabSelect Prisma, the protein A base. It's going to bind the FC part. It's going to also have interaction to VH3, so the heavy chain, on the orange part. And then we have our VL resin that is going to bind specifically to the left part, the kappa LC. And then I've thrown in one additional resin here, and that is MabSelect VH3. So that's going to bind selectively to the orange part. And then, of course, we're going to have 2 different antigens, the black and the orange. So here we did capturing with MabSelect Prisma, just to see, okay, we know we're probably going to capture the most impurities with this to characterize the start material, because there's a value in doing that. And then we did a gradient elution, lowering the pH slowly with VH3 and VL, because we thought they have higher selectivity, so they're potentially going to capture fewer impurities. And this is exactly what we're seeing. So if we just look, now there's a lot of numbers here, but we can just look, okay, if we capture it with Prisma, we don't aim to any separation here. We know that we have the correct bispecific. We also have homodimer, so black-black, half antibody. We have heavy chain and we have light chain, so we have a lot of impurities in this one. If we look at the MabSelect VH3, this one is going to fish out the correct one and more or less only the correct one. If we look at the VL eluate, here we're going to fish out the correct one, and then we can separate away, at least partially, the homodimer eluting later, and then dimer of the light chains that are eluting at the latest. We did a bispecificity on this as well and can see that we have then the correct binding of this construct. All right, I think I'm good on time. So I have a few more slides, and I'd like to end with another analysis method, because what I've shown you so far has been based on RPC. Most of it is intact mass. Now I'll show you a little bit about native SEC-MS. So there is a presentation I did in the spring. It's available on demand for the method development around this. It was pretty extensive, so I'm not going to go through it here, but I'm happy to take questions, or you can ask me later. But what I do want to show you is some updates and just some tips and tricks based on this. So the whole idea with using native SEC-MS is to speed up. So we want quicker time to results. And this is because we're combining SEC-HPLC, where we get the purity, with LC-MS, where we get the molecular weight. So ideally, we want to have the purity and molecular weight within the same injection. And this is exactly what we get. So here's just a reference method. It's our own column, an optimized phosphate buffer. It's one sample. You can see we have aggregates and main peak. We take the exact same sample, and then we analyze it on an XBridge Premier Protein Sec from Waters on a BioAccord system. And now we can't use phosphate, of course, because we're injecting it to the mass spec, so we use ammonium acetate. The mass spec data is not going to look as nice as an RPC, but it's sufficient enough to get the correct molecular weight to the right. Then we use the UV to assess the purity, and I hope you can appreciate that the purity profile with SEC-HPLC and native SEC-MS is going to be very similar. Challenge here, since we're limited in using ammonium acetate, is to get really nice separation and resolution between our peaks. And it can be very challenging to have the monomer and low molecular weight separation. This is done manually, so here we can select where we're going to split the peaks. It's pretty easy to detect this shoulder and assess the low molecular weights. What we're using on the BioAccord is automatic integration, and it's very challenging for any software to pick up that shoulder. This was not possible in the Intact Mass app version 1.4, but it is in 1.7. So this is the exact same sample, and here it can pick up this very challenging shoulder. And why? This is because Water has now implemented a thing called detect shoulders in the software, and then it will pick up these challenging integrations. So I hope you can appreciate that the purity profile we get with these two methods are very similar. Now I will end with just two tips and tricks. To get really nice data with native SEC-MS, for the separation in the LC, you need to have pretty high ammonium acetate concentration. If that doesn't work on its own, you need to throw in a little bit of organic content, for example, acetonitrile. So if we look at the blue trace here, this is going to be ammonium acetate 100 millimolar. This is probably where you're going to start. And then if we look at the most extreme case, so I've doubled ammonium acetate concentration and also added 2% acetonitrile. We're going to look at the red trace. Then we can see that we have better recovery of high molecular weight, so the really big one. And we also have better recovery of the high molecular weights. And this is still mass spec compatible. But increasing the ammonium acetate concentration and also adding organic content is going to suppress your mass spec signal. So one way to improve the mass spec signal is to drop the flow rate. We're typically running at 0.2 milliliters per minute, and here I dropped it to half, so 0.1. And this is going to increase your mass spec signal about 2-3-fold, and it's also going to improve your separation. So this is really a win-win. Here I'm just showing it's 10 injections overlaid. You can't really see them, so it's very, very reproducible. And we can see then the very high molecular weights, monomer, and then two different low molecular weights. So it's a very nice chromatography. This is my final slide, and I just want to add with that there are a lot of new antibody variants coming up. This is a very nice review highlighting that we can't think in the same way when we're going to purify and analyze them. I have highlighted throughout the talk that we need affinity chromatography resins that are selective for capturing MABs, because that makes the purification process much easier. And then, of course, we also need accurate analytical methods, for example, mass spec, to characterize these pretty challenging antibodies. And then my last slide, well, my last point here is just native-seq-MS. I think this is good because it enables quicker time to results, because you get both purity and mass determination within the same injection. So thank you very much.